This is group 3 and this is our oral discussion in chapter 4.
Case study 3 RNA enterprise RNI to control enzyme pests in cotton. Here's the context. Enzyme pests damage cotton crops. RNI technology silences genes in pests by feeding them plants containing double-stranded RNA sequence that match insect genes. Biochemical factors involved. First, RNA molecules are used to block a specific gene expression in target pests. Second, water as a transport medium for RNA molecules in plants. Third, soil buffers maintaining plant health and RNA active. Here's the challenges. Enzyme-resistant development over time. Second, ecosystem disruption. And here's the discussion questions. First, how do nucleic acid RNI help control pest populations in cotton herding?
So number one, nucleic acid RNI help to control pest population in cotton farming by reducing essential gene in target insect, by reducing our survival to our reproduction. When pests consume cotton plant, it engage double standard RNA.
Okay, let's talk about environmental analysis benefits. First, reduce reliance on chemical pesticide, lowering soil and water contamination. Second, target specific pests and minimizing harm to beneficial insect. And lastly, support sustainable pest control through biological means. And next, let's talk about challenges. First, pesticide may be develop resistance over time, requiring a new RNAi strategy. And second is potential unattended on effect on non-target species and ecosystem. And lastly, RNA stability difference condition, example soil and water, may impact effectiveness.
Second, how might soil pH and water balance affect the delivery of RNA molecules within the plant?
So, soil pH influence nutrient availability and RNA stability in plants. So, soil balance affect RNA transport moisture to ensure improper movement through plant and vascular system.
For environmental analysis, soil nutrients, soil buffers, regulate pH, impacting RNA efficiency. And the second, water availability. RNA may affect non-target organism. And the third is impacting or ecosystem impact. May lead for the last is gone. Resistant rest may lead and to the insect adaptation.
Third, what are the possible ecological consequences of pest population suppression using RNA?
Possible ecological consequences of pest suppression using RNAi. Number one, non-target effects. RNAi could unintentionally affect beneficial insects, including pollinators and natural predators. Number two, disruption of food chain. A decline in pest population may result food source for insect-eating birds, amphibians, and other organisms. Number three, resistant development. Pests may evolve over time resistance reducing long-term effectiveness of using RNAi.
So, soil and water contamination. So, soil and water contamination, degraded RNA molecules may persist soil and water. Soil and water. Soil and water contamination. Soil and water contamination, degraded RNA molecules may persist soil and water, potentially impacting microbial communities and non-target organisms. Then, ecosystem imbalance. Ecosystem imbalance may surface one fish species might allow other fish to proliferate, leading to unforeseen ecological shifts.
So now let's tackle about the environmental analysis. When we tackle about environmental analysis, we expect the positive and the negative impact of it. And the positive impact is, first, we can reduce in using chemical pesticides, and second is minimize soil and water pollution. And when we tackle about the negative impact is, the negative impact can potentially unintended consequences when it comes in our diversity that affected the food web and the ecosystem stability. To resolve this negative impact, we can lessen or reduce the use of chemical pesticides and to mitigate the negative impact into our environment. Thank you.